Hello, everybody. I am Nadine Rouphael, a professor in medicine at Emory University and the executive director of the Hope Clinic, the clinical arm of the Emory Vaccine Center. I am glad to present early data from the COVAIL trial, the COVID-19 Variant Immunologic Landscape trial, a work that has been funded by NIH with Angela Branch from Rochester University as the co-chair. This randomized phase 2 trial evaluates the breadth of antibody response after different vaccine variants, monovalent and bivalent, prototype, and variant. We have 3 stages in COVAIL. We have a stage 1, which is the Moderna stage, the stage 2, which is Pfizer stage, and the stage 3, which is the Sanofi stage. We also have an additional stage 4 from Pfizer that we are currently enrolling for. For the Moderna, each arm had a total dose of 50 micrograms vaccine, the approved Moderna booster dose. The arms were the following: prototype vaccine given as a single dose to 100 participants, Omicron and Beta given as a single dose to 100 participants, Omicron and Beta given as 2 doses separated by 56 days to 100 participants, as well as Omicron and Delta, Omicron and prototype, each one given as a single dose to 100 participants. The participants were adults 18 years and above, and they had received any combination of EUA or approved primary vaccine series and a single boost. They could have any infection status, either positive by an antibody, or they stated to us that they had a history of COVID-19. The study was in the US, including 22 sites, and then the primary vaccine series and the single boost were within the interval as stated in the CDC clinical guidance. We really wanted our study to reflect the US immunologic background. So for the Moderna stage 1, we enrolled between March 30, 2022 and May 6, 2022, 602 participants, 597 were vaccinated. And then we completed that enrollment in roughly 5 weeks after a study pause for a suspicion of myopericarditis. However, the cardiac MRI and the troponin I were negative. We also deliberately slowed enrollment to ensure recruitment of older adults, recruitment of adults with prior infection with a focus on diversity. So this is the summary of demographic and baseline characteristic by vaccine arm for the stage 1 Moderna. As you can see, we had good diversity. 8% were African-American, 82% were white. We also had 35% that were 65 years and above. We had 53% that were females. And in addition, we were able to meet the mark of 20% having prior infection by history or by an antibody. It's not a surprise that actually 95% of our study population received mRNA product prior to study enrollment. We allowed participants in the study if there was at least 16 weeks since last vaccine or dose. The median interval since last antigen exposure was 24 weeks. All these demographics and baseline characteristics did not vary between arms. So while today the focus is actually on the antibody data from 2 labs with pseudovirus titer, those labs were the Monogram lab, as well as the Montefiore lab at Duke University. We also collaborated with Derek Smith from the SAFE program. I just want to touch base just a little bit on the safety and the reactogenicity of the different vaccine candidates before I will focus on the immunogenicity data. So this is to look at the safety and reactogenicity. This is the local reactogenicity, which was mostly mild to moderate and only occurring shortly after vaccine. There were very few cases of severe local reactogenicity. This is on the systemic reactogenicity, whether they developed a fever, they felt feverish, they had fatigue, myalgia, arthralgia, headache, and nausea. And you see the same trend, no difference between the arms, mostly mild to moderate and then self-limited. We also had 4 SAEs in 3 arms deemed unrelated to study product, including 1 death. There were 4 adverse events of special interest that were reported, one of which was related to study product and has resolved. This was a male in his 30s that reported chest pain 1 day after vaccination that was evaluated as possible myocarditis and ultimately ruled out with a negative troponin I and a normal cardiac MR. So now, again, the focus will be on the immunogenicity data. Since 80% of the participants were uninfected, the focus of the presentation will be on this subgroup. The early response between young and older adults are similar and are combined in this presentation. And also, since 1 dose and 2 dose beta and BA1 bivalent vaccine arms are the same at day 15, they were also combined. Here we present the day 15 GMT and GMR to baseline for each arm and for each variant. So I'm showing you actually what are the titers to the D614G that's included in the prototype vaccine. And you can see that with every strategy tested, we saw an increase in antibody against D614G, the most, with the ID50 varying between 16,000 to 22,000 using the monogram lab. For Omicron BA1, day 15 GMT, the Omicron-containing vaccines had the highest GMFR compared to prototype, with an increase anywhere between 16 to 22 times, compared to 12 times alone for the prototype arm when compared to baseline titers. And as expected, the titers against BA1 were lower than D614G. Again, this is very early day 15 data. What we know from other IDCRC study, the mix and match study with Dr. Like and Dr. Atmar, that boosting with prototype vaccine increases level to D614G and BA1 very well early on. But there is a rapid decay over time within 3 months, particularly for the Omicron titers. So what do we know about variant vaccines and durability of antibody response? Here is the preprint from Moderna beta bivalent vaccine with a more durable response for the antibody titers that was noted against both the ancestral as well as the beta strains 6 months after the boost. This is the data from a study that we have conducted at Emory along other sites, the GMID 21002. So let's zoom on the bivalent prototype and beta with a bit more durable response at day 181 against Omicron BA1 than the prototype vaccine. So we are eagerly awaiting data on later time points to better understand durability of the variant vaccines we tested within our study COVAIL. Another way to look at the data is to look at geometric mean ratio using ANCOVA model. So all Omicron-based vaccines were compared to D614G. These vaccines had a geometric mean rise to Omicron BA1 greater than 1.5 relative to the prototype vaccine, with an unadjusted lower bound confidence interval of above 1, as you can see in the red box. This is quite similar to the data shown by Moderna on the bivalent Omicron prototype vaccine of a GMR of 1.75, 29 days after vaccination. Now to the Montefiore data. Again, I want to emphasize that the data is not yet complete, but we have here data on D614G, Omicron BA1, Omicron BA2121, and Omicron BA4, BA5, that's currently the predominant strain in the US. And that's for 4 different vaccine arms: the prototype, Beta Omicron, Delta Omicron, and Omicron alone. And here again, it's on uninfected participants. You note the lower titers for the Omicron subvariant, particularly for BA4, BA5, roughly a third of the titers that are observed for BA1. Again, hard to conclude with the partial data here. This is, though, similar to the data when we compare to recently published data from Moderna on their bivalent Omicron prototype vaccine, here against BA1 at day 29, and here against BA4 and BA5 at day 29. Now, more on antigenic cartography and antibody landscape. Those are common visualization tools used in the update of influenza vaccine formulation. Antigenic cartography uses antibody neutralization data to position virus variants and sera relative to each other. So the lower landscape and line correspond to the day 1 immunity. This is prior to them receiving a second boost within the COVAIL study. The upper landscape is the day 15 immunity. You see the titer plot shows GMT with a 95% confidence interval, where the superimposed blue line shows the prototype GMT in each panel. So you can see at day 15, a serologic advantage again for Omicron-containing vaccine, particularly against BA1. Here you can see the day 1 and day 15 landscape for uninfected and previously infected. The responses are better for previously infected, and basically day 15 for uninfected is the equivalent of day 1 for previously infected. You can also see that the baseline landscape is tilted towards D614G, while after vaccination, there is more of a flattening of the landscape compared to pre-vaccination for variants of concern. And you see here the beta on top, the delta at the bottom, and the BA1 on the right. So in both cohorts, Omicron-containing vaccine actually lift the titers against BA1 the most. So based on titer provider as well as other data, situating BA4, BA5 on the map was an important deliverable for our study, the COVAIL study. It tentatively falls even further than Omicron and Beta. And as you know, based on the FDA recommendation from a few months ago, the vaccine that's currently being used is a prototype component to increase the antigenic landscape near D614G, in case the new variant of concern will emerge in that part of the antigenic map, and also to boost the antigenic SIN, and a Omicron component to increase the antigenic landscape near Omicron BA1 and BA4, BA5. The all strategy tested and reported here provides suboptimal coverage of the antigenic space near BA4, BA5. The FDA was clear that the bivalent that's currently being used will contain prototype and BA4, BA5, where here we tested in this study a different combination, but then the Omicron subvariant we choose was a BA1. So in conclusion, all strategy tested increased titers to Omicron BA1 at day 15. Of combination that have been tested, all arms cover BA4, BA5 similarly at day 15. The limitations of our study is the small sample size, not allowing further stratification based on prior vaccination, though the majority had received the mRNA product, or interval since last antigen exposure. We yet don't have access to cellular or innate immunity data, but we have to have it available soon. And then higher vaccine doses were not tested. We used the exact same amount of antigen that are used for a regular booster. For Moderna, for example, it was 50 microgram. For Pfizer, it was 30 microgram. Data on future time points and stages are pending. Our next stage is currently testing BA4, BA5 with prototype vaccine candidate versus BA1 plus prototype vaccine candidate that's used in Canada and the UK. We should have data on day 29 and day 91 available soon. We also should have data 6, 9, and 12 months later for all stage 1 participants, as well as other stages, and against D614G, Beta, Delta, BA1, BA4, BA5. We should have also the cellular data coming up soon. This will allow us to understand better the correlates of risk and protection for Omicron wave, as we had many cases of COVID during the period of enrollment and follow-up. This is a huge effort done by 22 sites across the US, funded by NIH, and the support of 3 labs, and also many that had supported the operation aspect of COVAIL. I really want to thank the Division of Microbiology and Infectious Disease. I want to thank the SAFE program, the Infectious Disease Clinical Research Consortium Leadership Group, and the IDCRC Laboratory Operation Unit, Derek Smith's lab that's based in Cambridge, UK, David Montefiore's lab based in Duke, North Carolina, FHI 360 that provided valuable support for protocol development, MS that provided statistical and data management, also PPD and Leidos for their support of the operation, and most importantly, our participants. Without them, science could not move forward. Thank you so very much for paying attention to the talk. This will conclude the talk.